All right, biologists, let's talk about proteins. This is Theme B, form and function, topic B1 molecules, subtopic B.1.2. So let's start by talking about the generalized structure of an amino acid. To draw an amino acid, you have to start with the alpha carbon. The alpha carbon has this name because it's connected to different things, 4 different things. So this here is our alpha carbon. It is connected to the amino group, and the amino is NH2. It is connected to the acid part of amino acid. But remember that carbons must make 4 bonds. Here we have 1, 2, 3, 4, but here we only have 2. What are the other 2 bonds? One of them is a simple hydrogen, and the other is what we call the R group. This R here can be represented by many different things. So let's check. We have our alpha carbon, the amine group. You don't have to draw it like this. You can draw NH2, but you have to connect nitrogen to carbon necessarily. We have the acid, we have our hydrogen, and the R group. There are 20 different R groups, and those different R groups that characterize different amino acids. It's like letters of the alphabet. We have different 20 letters to create all different words that we have. We have 20 different amino acids that can be combined in many ways to create all the different proteins that we have with all the different functions that they have. And how do we combine these amino acids? By a type of reaction called condensation. And this condensation reaction forms peptide bonds. So here we have 2 amino acids. So we have amino acid 1, amino acid 2, and they are close together. You have the acid part of one amino acid, the amino part of the other amino acid. And look what's happening here. You have OH. You have 2 hydrogens, 1 oxygen, H2O. So the condensation reaction will happen when this H2O forms, this water molecule forms, and now you have the carbon here needs to make a bond because now it has 1, 2, 3 bonds. Nitrogen here needs 1 more bond because it lost the hydrogen, right? So we have this comes out. So you have H2O here, and now you have the other 2 amino acids missing parts. And what do? The carbon needs to make a bond. Nitrogen needs to make a bond. We make a bond between the 2 of them. This new bond that did not exist before, that now is uniting the 2 amino acids, this is what we call the peptide bond. You can continue this reaction. You keep adding more amino acids, and then you have a longer chain of amino acids. You have a protein. The organelle that does that is the ribosome. Now, we know there are 20 different types of amino acids, and from a dietary point of view, we can classify them into essential and non-essential amino acids. Essential amino acids are the ones that you need to ingest. You cannot make them. Now, let's be clear. It's not like we can make amino acids from the absolute scratch. We still need protein in our diet. We still need amino acids in our diet, but we can convert some amino acids into others. The amino acids we can make from other amino acids are called non-essential because they're not required. It's good to eat them. It's good if you eat them, okay? But in case you have a deficiency, you can use another amino acid and modify the other amino acid to make a non-essential amino acid that is lacking in your diet. But the essential amino acids cannot be made by you. You need to eat them. Right. So if you're vegan or plant-based diet or, strictly vegetarian, no sorts of animal protein whatsoever, you need to take some care with this because plants are not as rich in amino acids, in proteins, as animal sources of food are. However, it's not to say that you cannot live exclusively on plants. You can meet all your protein requirements from plants. Ideally, you would associate a cereal like rice with a pulse like beans or soy or lentil because cereals lack certain amino acids, but pulse have them in a good amount, and vice versa. The amino acids that pulse are lacking, cereal are rich in those. So they complement each other really nicely, like yin yang, but made with rice and beans. Now, if you consume animal protein, you don't need to worry about essential amino acids. You surely are getting your, enough amino acids from your diet. So can you give me an example of a non-essential amino acid? Sure, hydroxyproline is actually not one of the 20 amino acids. It's kind of like the 21st amino acid. We make it from proline that we get from our diet, and a special enzyme will take proline and vitamin C and turn that into hydroxyproline, which is super important for making collagen. Now, you might have heard at some point that the lack of vitamin C leads to scurvy, and this is because you need vitamin C as well as proline so that enzyme can make hydroxyproline, which is necessary for collagen. Scurvy is actually a collagen disease. It's a deficiency of collagen. You cannot make collagen because you don't have vitamin C, which is required to make hydroxyproline. And then you tell me, well, 20 amino acids doesn't seem all that much. How many proteins can we make? Well, think of this way. Imagine if you had a chain of only one amino acid, which is just one amino acid. How many possible combinations of one amino acid you can have? Well, there are 20 different amino acids. So 20 to the power of one amino acid here, you have 20 possible combinations. With two amino acids, the number of combination jumps to 400. I'm going to let you pause here the video and try to fill out this table. Okay, are you done? Let's check our answers. With 10 amino acids, you can make more than 10 trillion combinations. And 10 amino acids, not even a protein. 10 amino acids is short. It's extremely short. So the immense variety of life there is arises from the infinite variety of possible amino acid, of possible proteins that carry out all sorts of functions because they have all sorts of forms. Here we see a generalized peptide chain. We see the alpha carbon here in blue, the acid part, and in red, we have the peptide bonds. So what do we do with all these different peptide chains? What do we do with all these proteins? There are many roles that proteins occupy in a cell or an organism. Probably most famously, enzymes. Enzymes are incredibly important proteins. They catalyze reactions. The most important, the most common enzyme in the world by mass, if you take all the enzymes, all the proteins, and say, okay, what do we have the most? Is an enzyme called rubisco, which is part of the photosynthesis reactions, and every plant has them. Every plant out there doing photosynthesis had tons of rubisco. But not only that, hormones. Some hormones are lipids, but so many others are peptides. Like insulin, for example, very famous hormone. Insulin is a peptide, so it's made of amino acids linked by condensation forming peptide bonds, and it is released after a meal to let glucose into the muscle cells, into the adipose cells. What else? Immunoglobulins, the famous antibodies. They are also proteins. They're there defending your body against infection. They're part of the specific immune response. They play a critical role in our health, and they are also made of amino acids. Some proteins are pigment proteins. They interact with light, like rhodopsin in your eyes, that can interact with light and tell your receptor cells if you are receiving light or not. Or yet, hemoglobin in your blood is not made to interact with light, but it's a pigment because it's red, and it is crucial because it transports oxygen. And of course, as you might have heard in middle school, proteins are important to build our bodies. Well, there are structural proteins as well. Like we saw, collagen is a super important structural protein in animals. It's part of the connective tissue. It's part of bones, cartilage, ligaments, and they can even be used to form structures for the organism. Like spider silk is also made of proteins. Now, we have to know the effect of these two factors on the structure and therefore the function of proteins, pH and temperature. Every protein has their specific pH and temperature in which the shape is ideal. Therefore, the protein is performing their function as its best, right? This is called the optimum pH and optimum temperature. Now, the proteins can lose its form permanently, and therefore they completely lose their function. This process is called denaturation, and you've seen it if you ever cooked or fried an egg. The egg white, when it's raw, it's transparent, it's fluid, it's a viscous fluid. But if you cook an egg, if you boil an egg, for example, the heat will make albumin, a protein in egg white, lose its form. And that's permanent. You cannot unboil an egg. It is, once you boil an egg, it's forever. You cannot reverse this process, right? So this permanent loss of structure is called denaturation. You are denaturing the protein. And this change is so significant, it's so impactful that even the physical state and color of egg white change. You go from a clear viscous liquid to a white solid. So the effect of temperature is excessively high temperature will denature a protein, but low temperatures do not denature proteins, right? Low temperatures will decrease the rate of enzymatic reactions, will make enzymes work more slowly, but they will not permanently alter the shape of the protein. Excessively high temperatures, on the other hand, are dangerous because they will change the shape of proteins and therefore you lose function forever. This is why fevers, extremely high persistent fevers, can be dangerous because you are submitting your body to higher temperatures than what it's made to endure, to survive. As with pH, too much acid or too much alkaline, right? If a pH is too acid, lower than the optimal pH, or too alkaline, higher than the optimal pH, then it will denature the protein. Now you have to keep in mind that the optimum pH or optimum temperature depends on the protein, depends on the organism. A protein that exists in your stomach, for example, which has a very acidic environment, is made to function at a low pH. It will not work well in your mouth where the pH is 7. And vice versa. An enzyme in your mouth does not work in the stomach because the pH is too low, right? And the same can be thought of temperature. There are some archaea, some microorganisms that only live in volcanic lakes where the temperature is super high. For them, 37 degrees is mortal, is absolutely, it's fatal cold. For them, they cannot survive the cold of 37 degrees because they're made to work in environments of like 80 degrees, 90 degrees. Our temperatures would denature in such environment. But for those archaea, that's the optimum. So there's no universal answer. What is the optimal pH? What is the optimal temperature? It depends on the context. It depends on what you are talking about.